Alright, I want to take a little bit more time here and in this video tell you about DNA in a little more detail. Now, if you remember, DNA, of course, has three chemicals that come together in a linear form. So those are adenine, cytosine, guanine, thymine, and then if we're talking about RNA, we replace the thymine, the T, with uracil. So in RNA you use four chemicals also, and they're the same, but instead of the thymine we use uracil, so we represent it as a U. I will probably never ask you directly what the A, C, G, T, or U stand for, but you do need to know that these are the four chemicals that are used to make either DNA or RNA. And you need to understand that these chemicals bind to one another. Alright, so in this particular strand of DNA, we have A bound to G, bound to C, bound to T, bound to A, bound to C. Now this is a single strand of DNA. Now we could have a similar strand down here of RNA. I'll just make one up. Let's go. Oops. What? You can't have T's in RNA. Can't have E's in either one. Let's see here. Alright, so here we go. That would be like an RNA, and this would be single-stranded DNA. Now we also know that DNA typically is not single-stranded. It's going to have what's called a complement to form the double-stranded helix structure that we've seen in DNA. So we would have to represent the complement, and you need to know how to do this. And to create the complement, we know that whenever there's an A on one side, on one strand, we know that on the other side it's going to be a T. And vice versa, whenever you have a T on one side, you're going to get an A. And wherever there's a C, there's a G. And of course, if there's a G, you're going to have a C. So, in other words, we need to come down here and create what we call the complement to this strand. And so here is an A, so we know that we're going to get a T on this side. And here is a G, so that's going to be a C. And there's a C, so that's going to be a G, an A, a T, a C, a G, three A's, two T's, a G, a C, a G, an A, three T's, a G, C, A, G, G, T. So this would be our double-stranded DNA. We would have one strand, alright, with this sequence, and the opposite, the complementary strand, would be this one. So understand that this is how DNA works. It has strands, and they are two complementary strands. They are complementary, that's the way we say this. They are complementary to one another, and this is how, if you know one strand, you can figure out the other strand. So that's DNA, alright. We also need to understand that we're going to read, alright, one of these strands, and we're going to make an RNA out of it. So eventually we're going to be reading a strand, and when we read it, we will create the RNA, and the RNA will be the complement. So in this particular case, if we were to make an RNA of, let's just take part of this. Let's say we took this strand right here, and we make our own RNA complement, which you will have to know how to do eventually. So we're going to start with these three T's and go to this first A. So here's a T. If we're starting right here, alright, then what's the complement? Well, over here it's A. Well, RNA also uses A, so we would say that's an A. So we're going to get three A's, but then you get an A, and normally that's a T, so we're going to put U's, and then we're going to get G, and C, G, and that's a T, so it's an A. Oop, I put a V in there. GCG. There we go. There's an A and then a U. So realize that the RNA complement is very similar to the DNA complement. If you'll notice, this piece and this piece are almost identical. All we did was replace what? We replaced the T's with U's. So we make RNAs, we make these copies in order to utilize the information in the DNA outside of the nucleus. So remember that we're storing this DNA in the nucleus to keep it protected so it doesn't get damaged. And to use the information, though, we have to somehow get the information that's in the DNA outside of the nucleus. So what happens is you make a RNA copy, and then you send that single-stranded RNA copy outside the nucleus. So if you want to think of it this way, okay, here's our cell, and here's our nucleus, and in the nucleus I have the DNA, and the DNA is usually in the form of a chromosome. All right, and so here's our chromosome, horribly drawn there. And a little piece of that chromosome is this sequence right here, all right? And what will happen is when you want to utilize the information, you will actually force the DNA, which is kind of like this. Let's say this is our sequence there, all right? We kind of see this as being the equivalent of this here. What we're going to do is we're going to unzip the DNA briefly. All right. Let's see if I can draw this without making a mess. All right. And so here is the part that's zipped. And here's the part that we have unzipped. Right here. So we have the unzipped part down here, and with that, once it's unzipped, proteins can stick to it, and when they do, they can read it and make copies. I'll have another video talking a lot more about this, but what I want to talk about right now is the fact that when you unzip it, the DNA is single-stranded, and RNA is always single-stranded. Your body doesn't like double-stranded RNA, and you have proteins that will attack it and destroy it because there are viruses that are typically double-stranded RNA, so your body just recognizes double-stranded RNA as a bad thing, and it goes, attacks, and destroys it. But in the DNA and the RNA, when it is single-stranded, there can be the formation of something, and these will be very important in the next video, or in a future video, so I want to tell you about them. If, for instance, I get, let's make a new document here. Let's say I get let me make this big. All right, let's say I get a sequence, AGC, something like that. But let's go on a little bit more. And let's say this is the single-stranded part, this region down here. So this region down here has this sequence. Well, if you'll notice, I've written this in a very specific way. I have a lot of T's, followed by a couple of GC's, G's and C's, and then a whole lot of A's again. What will happen is if you have this sequence, all right, it's going to form what's called a hairpin loop. All right, and the A's and the T's, see this linear piece of DNA can kind of flap in the wind like a flag maybe. So it's going to be in a liquid environment, so it's going to be moving around as the liquid moves. And what will happen if these are single-stranded, this piece here, the part with the T's may be able to come in contact with the A's. And what did we just learn? We learned that what? A's bind to T's and C's bind to G's. Well, the C's and G's are all cramped up in here. And it's not enough room. These are flexible, but they're not completely flexible. And there's not enough room for these to contact one another. But there is enough room for these, as these DNA is kind of moving around, to perhaps come in contact with these. And that forms this thing called a hairpin loop, and it looks like this. Alright, and then here's the rest of the DNA. So along here, you have your A's and T's. Up here are your G's and C's. So the G's and the C's are going to be in this region. And then the A's will all be in a line here. And the T's will be over here. Alright, up and down here. Or vice versa. I'm sorry, I have that backwards. We started with the T's. So the T's would be over here and the A's would be here. Let's see if I can undo that. Actually, I think there's an eraser on here. I should probably use that. But undo is pretty simple to use. Alright, so your T's will be over here. And your A's will be here. And your G's and C's will go up here at the top. These will not bind to one another. They will just make this loop. But the T's and A's here that I have, as you can see, they're going to come together and form this little loop. And this is going to be very important for some of the things we talk about. There's going to be something called a terminator. And the terminator is very important for us in defining how genes work. So understand that this little piece of DNA, alright, might be here, it's going to be at the end of a gene, is going to be on this chromosome, alright, and so the gene itself is just a small piece of the chromosome. Not all of the DNA, there might be some more DNA out here, alright, is going to make up the genes. The genes are just a small part of the DNA, alright, so you might have lots of DNA that are all wound up into this chromosome, but only a small part of that DNA is going to be a gene. And what we're going to find out is that there are places that are genes and there are places that are not genes. And the vast majority of the DNA are not genes. It's actually a very small percentage of the actual DNA that are genes. And the genes, of course, are instructions for making proteins. So I just wanted to introduce this to you and show you the idea of a hairpin loop. Now you can have hairpin loops with DNA or RNA, and this thing called a terminator can be found by looking at the DNA or the RNA, but as a general rule, we just need to know about hairpin loop.